Hello friends, in this video we are going to take this topic of enzyme inhibitors, which is very important, especially questions on competitive and non-competitive inhibitors are frequently asked about Km and Vmax, what happens, right? So let's do this topic conceptually. So here we are going to take 6 inhibitors. First, we will be taking competitive inhibitor, then is non-competitive, then uncompetitive, allosterically inhibitor, which I have already covered in regulation also, then suicidal inhibition and feedback inhibition. Okay, so let's start with this. So the first one we'll be doing is competitive and non-competitive. Tell me, for any competition, what is the basic necessity? For any competition, things must be similar. Only then a competition can occur. For example, there cannot be any competition between a 4-year-old child and a 50-year-old adult. Okay, things should be similar for a competition. So, in case of competitive inhibition, the inhibitor resembles in structure with the substrate. Inhibitor resembles the substrate. Substrate binds at active site. So, inhibitor also, in this case, inhibitor also will bind at active site. Okay. Now, before moving for the detail of competitive inhibition, first let's do the revision of Km. What is Km? Km is a constant. It is Michaelis-Menten constant. I already told you. And I told you that it does not depend on change in substrate concentration or change in enzyme concentration. It is a constant value, right? It will not change with change of enzyme concentration or change of substrate concentration. But Km depends on affinity, and you know that Km is inversely proportional to affinity. The affinity, the attraction between enzyme and substrate. Okay, Km is inversely proportional to that, and Km defines the affinity between a particular enzyme substrate pair. For a particular enzyme substrate pair, Km is a constant value, it will not change. Okay, but in case of competitive inhibition, the affinity between enzyme and substrate decreases because now enzyme has affinity for inhibitor also. Now, in case of competitive inhibition, when competitive inhibitor is also present, then competitive inhibitor is also present, so the enzyme has affinity for substrate also, but it also has affinity for the inhibitor also. So the affinity between enzyme and substrate decreases. So what will happen to Km? If affinity decreases, means Km will increase. So Km will increase in this case. In case of competitive inhibition, Km increases. Otherwise, normally it is a constant value. It will not change. Okay. So let's see the detail of competitive inhibitor. Look, this is the enzyme. This is enzyme. This is active site of enzyme where substrate binds. So this green colored substance here which I am showing is substrate, and this brown color is inhibitor. You can see that both have similar structure. Okay, so substrate will bind at active site, inhibitor will also bind at active site, right? Now, if substrate binds at active site, then enzyme substrate complex will be formed. If inhibitor binds at active site, then enzyme inhibitor complex will be formed. Okay, but in this case the affinity is decreased. Affinity is decreased, so Km is increased. Km means substrate concentration. Km is nothing but substrate concentration. So substrate concentration is increased, right? When substrate concentration is increased, then there are more chances that out of substrate and inhibitor, substrate will bind at the active site, making enzyme substrate complex. And enzyme inhibitor complex is not allowed to be formed. See, there's a competition between these two. Okay, in a competition, whosoever is more, that will be the winner. Okay, so here when substrate concentration increases, then substrate is more, so inhibitor's effect is nullified. Okay, the inhibitor is unable to act on the enzyme because randomly there are more chances that substrate will bind at the active site. So when always substrate is able to bind and inhibitor is not allowed to bind, then with this inhibitor velocity does not decreases. Look, inhibitor means it will decrease the velocity now. So here velocity is not allowed to be decreased. Why? Because Km is increased, because substrate concentration is increased, right? So let's read these points. In competitive inhibition, inhibitor and substrate resemble each other. Inhibitor binds active site. Vmax, velocity, is same. Km is increased. And in this case, inhibitor cannot bind with enzyme substrate complex. Let's say first substrate comes and binds the enzyme, making enzyme substrate complex. Now if inhibitor will come, will it be able to bind? No, because active site is occupied by substrate. So inhibitor cannot bind enzyme substrate complex. Inhibitor can only bind the free enzyme. Okay. Last point is it decreases the affinity. Okay, I told you already that affinity is decreased in this case. So we are done with competitive inhibitor. Next is non-competitive inhibitors. Now there is no competition between the two, between the inhibitor and substrate. So first point, they do not resemble in structure. And here you can see the enzyme is this. Enzyme is this. This is active site of the enzyme where substrate binds. And there's one more site here, that is regulatory site. At regulatory site, inhibitor binds. This non-competitive inhibitor binds at the regulatory site. It is also called allostric site. Okay, I told you. Now, I already told you for such an inhibitor, what happens that when this inhibitor will bind at the regulatory site, it will induce changes in the active site so that substrate cannot bind. Even if we have one molecule of inhibitor and 1000 substrates, still who will be the winner? Who will be the winner in this case? Always inhibitor will be the winner in this case, right? Because this substrate will not be able to bind. Once inhibitor is present, it has changed the shape of active site so that substrate cannot bind. Okay, so in this case velocity will be decreased. In this case Vmax will be decreased. Okay, in this case the affinity of active site of enzyme with the substrate is not affected. So affinity is not affected, so Km is also not affected. Km is unchanged, right? So I can say that this kind of inhibition is irreversible. If such inhibitor comes, we cannot reverse its effect. In your books, when you see, they say that non-competitive is further of two types, irreversible and reversible. But mostly it is irreversible. So that's why I'm just telling you this much. Okay, there's no need to go into unnecessary details. So you just learn this much, that non-competitive is mostly irreversible. On the other hand, we did for competitive. In case of competitive, we can say that if inhibitor first came and decreased the velocity, let's say first inhibitor came, competitive inhibitor, and it decreased the velocity, but after some time when the Km will be increased and substrate concentration will be increased, then velocity will be same. That is, we can achieve same Vmax which is possible without inhibitor, because we can nullify the effect of that inhibitor by increasing Km, right? So we can say competitive is reversible, but non-competitive is mostly irreversible, right? So let's read the points. Here inhibitor does not bind the active site, it binds at regulatory site. Vmax is lowered, Km is unaltered. Inhibitor can bind with enzyme substrate complex. In case of competitive inhibitor, I told you, inhibitor cannot bind with enzyme substrate complex. It can only bind with the free enzyme. But in this case, inhibitor can bind with enzyme substrate complex also. It can bind the free enzyme also, right? So how it binds enzyme substrate complex? Because it is not binding at active site. Even if substrate is present, substrate has occupied active site, but inhibitor can bind at the other site, which is regulatory site. So inhibitor can bind enzyme substrate complex. Then this inhibitor does not changes the substrate affinity for the enzyme. Then this inhibition is mostly irreversible. We have already done that. Okay. Now let's take the examples of competitive inhibition and non-competitive inhibition. So first we will be taking competitive inhibitors. Arsenate and oxamate, these two we did in glycolysis. Arsenate inhibits the enzyme glyceraldehyde 3-phosphate dehydrogenase. The detail of arsenic poisoning already I have told you in glycolysis. Then oxamate is competitive inhibitor of lactate dehydrogenase, right? So these two are inhibitors in glycolysis. Then next two are inhibitors in TCA cycle. Malonate, the 3-carbon compound, is competitive inhibitor of enzyme succinate dehydrogenase, right? Then fluorocitrate, C for citrate, C for competitive inhibitor. Fluorocitrate is the competitive inhibitor of aconitase. In non-competitive, next page we will do that fluoroacetate is non-competitive inhibitor of this enzyme aconitase. Okay, so these two we have done in TCA cycle. Next is sulfonamide. Sulfonamide is PABA analog. PABA analog. Analog means it resembles in structure. And what is PABA? PABA is para-amino benzoic acid. Okay, this PABA is required for the synthesis of folic acid in bacteria. For the synthesis of folic acid in bacteria, and this is a vital reaction of bacterial cells. And sulfonamide is competitive inhibitor here, inhibiting the formation of folic acid in bacteria, which is vital for them. So sulfonamide can be used as antibacterial. Okay. Next is methotrexate. Methotrexate inhibits enzyme DHFR, that is dihydrofolate reductase, which converts dihydrofolate to tetrahydrofolate, and this is a vital reaction in human cell. This is a vital reaction in human cell, and methotrexate competitively inhibits this reaction. So methotrexate can be used as anti-cancerous or immunosuppressant, right? Okay. Next is dicumarol. Dicumarol is vitamin K analog. It is vitamin K analog. It resembles in structure with vitamin K. Vitamin K is used in body for coagulation. It is used for coagulation. So dicumarol can be used as anticoagulant. Okay. So you have to learn that dicumarol is competitive inhibitor here, right? So all these examples are of competitive inhibitors. Next is about ethanol and methanol. This ethanol is used as treatment in methanol poisoning, because what happens in methanol poisoning, methanol is taken by enzyme alcohol dehydrogenase, and the product is formaldehyde. Formaldehyde, which is the toxic compound responsible for the poisoning effect. Okay. Now, in this poisoning, as treatment, we give ethanol. When both ethanol and methanol are present, then this enzyme alcohol dehydrogenase, because it has more affinity for ethanol, so it will do reaction of ethanol. Okay, so this enzyme alcohol dehydrogenase converts ethanol to acetate, and acetate is a non-toxic compound. So when ethanol is present, then this reaction will not occur, so the toxic formaldehyde will not be formed. So this is used as treatment in methanol poisoning, right? Then the last one is statins. Statin group of drugs, they inhibit enzyme HMG-CoA reductase. HMG-CoA reductase enzyme, which is rate limiting enzyme of cholesterol synthesis. It is rate limiting of cholesterol synthesis, right? So we have done all these examples of competitive inhibitors. Now next is non-competitive inhibitors. So first two we have done in glycolysis. Here we have iodoacetate, which is non-competitive inhibitor of glyceraldehyde 3-phosphate dehydrogenase, and sodium fluoride, which is non-competitive inhibitor of enzyme enolase, right? Then next two in TCA cycle, I already told you fluoroacetate is non-competitive inhibitor of aconitase. Then arsenate is non-competitive inhibitor of alpha-ketoglutarate dehydrogenase. Then cyanide is inhibitor in ETC, and it inhibits complex 4 of ETC, right? Then next two I am taking together, heavy metals and dimercaprol. Heavy metals and dimercaprol, they both inhibit sulfhydryl group. They both inhibit sulfhydryl group, which is present at the active site of many enzymes in the body. It is present at the active site of many enzymes in the body, right? Next is disulfiram, the Ent-abuse compound, disulfiram. This inhibits enzyme aldehyde dehydrogenase non-competitively. So be careful, in methanol poisoning, the enzyme which is responsible is alcohol dehydrogenase, but in disulfiram, it is aldehyde dehydrogenase. Okay. Then is diisopropyl fluorophosphate is the non-competitive inhibitor of all the serine proteases. Serine proteases have serine at their active site, okay, like trypsin, chymotrypsin, right? So we are done with non-competitive inhibitor and competitive. Next is uncompetitive inhibitor, also called anti-competitive inhibition. In this case, Km decreases, Vmax also decreases. So in this case, both Km and Vmax decreases. There is no need to go into the detail of this type, right? Just 2-3 points I will tell you. You can just note down that and just cram this. In this case, inhibitor can only bind enzyme substrate complex, right? Like I told you, competitive inhibitor can bind the free enzyme only. Non-competitive inhibitor can bind free enzyme also and enzyme substrate complex also. Now this uncompetitive inhibitor, it can only bind enzyme substrate complex, right? Here one example you can note down. One example of uncompetitive inhibition, acetylcholine. Acetylcholine inhibits placental ALP. Placental ALP. What is ALP? Alkaline phosphatase. Alkaline phosphatase, right? So we have done these three. So let's write down about Km and Vmax. What happens to Km? What happens to Vmax? In case of competitive inhibition, Vmax, Km increases and there is no change in Vmax. Non-competitive, no change in Km, but velocity decreases, right? In case of uncompetitive, both Km and Vmax decreases, right? So this thing is frequently asked, right? Okay, so we have to do, we have to do 6 types. So these 3 we are done. Even allostric we are done. Along with regulation, I have told you that allostric enzymes or regulatory enzymes, they have 2 sites. One is active site for the substrate, other is regulatory or allostric site for the regulator. Regulator can be activator or inhibitor, right? So whenever this allostric regulator binds at its allostric site, it will induce changes in the active site, so that substrate can bind or not bind, it can decide. The regulator will decide. That's why it's called regulator. Okay, regulator means control. Okay, so I've already explained allostric, but tell me one thing, that what is the difference between allostric and non-competitive? Because in non-competitive also I have told you similar things, in allostric also I have told you similar things. Then tell me what is the difference between them. Look, I'll tell you. Non-competitive, if you remember the examples of non-competitive, I have told you all the drugs. The examples are all the drugs coming in the body and acting as non-competitive inhibitor. Okay, but allostric is a way of regulation. It is a normal, natural thing in the body, that it is a normal way of regulation or control of enzymes and pathways, that we have allostric activators also, we have allostric inhibitors also, right? So allostric is a normal natural thing and non-competitive is drugs. Examples are drugs, right? So in order to explain better, I can just say simply that non-competitive inhibition is a topic in pharmacology mainly, right? And allostric regulation is a topic in biochemistry, in enzymes, that how enzymes are regulated. Okay, so I hope you can understand. So let's do the next one, the fifth type, that is suicidal inhibition. Suicidal inhibition, also called mechanism based inhibition. There is a proper mechanism for this inhibition. Let's take example, then you will understand it better. The word suicidal you can easily understand. So example is that allopurinol inhibits enzyme xanthine oxidase. Okay, this you know that this drug allopurinol is used in gout also. Okay, so how it is suicidal inhibition? Because allopurinol is not directly inhibiting this enzyme. What will happen, that allopurinol will act as substrate, will first act as substrate of the enzyme xanthine oxidase, making oxypurinol product, making oxypurinol. This oxypurinol, when it is increased too much, then it will inhibit the enzyme xanthine oxidase. So I can say that oxypurinol is doing suicide. Oxypurinol is inhibiting that enzyme which is making it, right, which is making oxypurinol. So this is a kind of suicidal inhibition, right? This oxypurinol is also called oxyprim or alloxanthine. Okay, so options can be there, so you can mark. So this is one example of suicidal inhibition. One more example, write down. Aspirin. Aspirin is suicidal inhibitor of cyclooxygenase, right? So we are done with 5 types. The next one is the last one, 6th type, is feedback inhibition. Feedback inhibition, also called end product inhibition. For example, cholesterol inhibits the rate limiting enzyme of cholesterol synthesis, that is HMG-CoA reductase. So for this pathway, it is a very big pathway, like around 20-30 reactions in this pathway of cholesterol synthesis. The rate limiting enzyme is HMG-CoA reductase. So the end product cholesterol inhibits the rate limiting enzyme. Okay, so this is called end product inhibition or feedback inhibition. Now, do you have any confusion between suicidal inhibition and end product inhibition? In suicidal inhibition also end product is inhibiting. In feedback inhibition also end product is inhibiting the enzyme, right? So what is the difference? Like similarly, the difference which I told you for non-competitive and allostric, here also it is the same difference, that suicidal inhibition or suicidal inhibition, that is drugs are in the body coming from outside, they act as suicidal inhibitors of the enzymes. But this feedback inhibition is a normal natural thing in the body occurring for the regulation of the pathways, for the control of the enzymes, right? So you can see the examples of suicidal inhibition, they are drugs, right? An example of a feedback inhibition is a normal pathway in the body, right? So this is it with this topic. Thanks for watching. Best wishes to you.